Professor Robert Langer to the Chemical Engineering Distinguished Seminar Series. As you guys may know, Professor Langer is really an inspirational scientist, an educator, and an entrepreneur, working and really pioneering the areas of drug delivery, tissue engineering, and regenerative medicine. So today is a sort of a special event. It's a slightly different type of distinguished seminar series. It's online, but what we're doing today is we're having half an hour talk by Professor Langer, and then half an hour sort of question and answer panel style discussion. So please think of questions and pop them in the chat at the end, and then we'll have a Q&A session. We've been sent some questions in advance, but if you guys have any further questions, do please pop them in the chat. So before we get started, a few words, further words on Professor Robert Langer. He really does need no introduction. He's an impeccable scientist with impeccable credentials, really too many to mention. Two things I'd like to mention is, he really is the highest, second highest cited H-index scientist of any individual in any field, and the highest engineer in history. He's also won multiple awards, including the United States National Medal of Science and the United States National Medal of Technology. And given that we're here in the UK, I should also mention he won the Queen Elizabeth Prize for Engineering. On the entrepreneurship front, his patents have been licensed or sub-licensed to over 400 companies, and he's the co-founder of a number of companies, including Moderna. So without further ado, I'd like to hand you over to Professor Langer. Let me just share the screen and the presentation. So window. Oh, sorry, I'm just.
That's the end.
That's the end. That's the end. Just go to the front. Okay, this you should all see the screen now. Okay, so Professor Langer, over to you.
Sure. Well, thank you so much. It's a pleasure to speak to you. I'll start right in and just go over how I got started. So I'm a chemical engineer, and I got my degree in chemical engineering from MIT in 1974. And that year, everybody that was a chemical engineer, pretty much like all my friends, went to work for oil companies. But I wasn't that excited about it. And so I started to look for I got actually 20 job offers, 4 from Exxon alone, and I wasn't that good, but they just had so many openings. But I just wasn't that excited about it. They were talking about increasing the yield of different chemicals by tiny percentages, and I just didn't want to spend my life doing that. And it made me think what I did want to do. And one of the things I did when I was a graduate student at MIT was help start a school for poor kids. And I got very involved in developing new math and chemistry curricula. And one day I saw an ad for that at City College in New York City to do just that as an assistant professor. So I wrote them a letter applying for a job, but they didn't write me back. But I really liked the idea, so I found all the ads I could to do that, about 40 of them, and I wrote them all, and I don't think any of them wrote me back. So that wasn't going so well. So I started to think, what else could I do with my chemical engineering education to help people? And I thought about medicine. So I wrote to a lot of hospitals and medical schools, and they didn't write me back either. And then one day I walked into the lab I was in, and one of the older scientists said to me, Bob, he said, there's a surgeon in Boston named Judah Folkman. And he said, sometimes he hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. So I wrote Dr. Folkman, and he was kind enough to offer me a job. If we could go to the next slide. And Dr. Folkman had this idea, which is shown on this next slide, that if you could stop blood vessels, you could stop cancer. And in particular, he said that the blood vessels, the reason they came in was because tumors secreted a chemical signal, which he called tumor angiogenesis factor. And that caused the surrounding blood vessels, which normally don't do anything, to start growing to the tumor. And he further proposed that if you could stop that from happening, maybe that would be a new way to treat cancer. I thought this was a wonderful idea. It turned out, I didn't know anything about biology. I still don't know very much, but I thought it was a great idea, but it turned out it was very controversial. Anyhow, he hired me, and my job, he said, was to prove he was right, and in so doing, isolate the very first substances that stop blood vessels. Well, one of the keys to doing something like this is having a way to study blood vessels, and there really weren't any good ways at this time, so we had to invent one. And if we could go to the next slide, what we began doing was using places like the rabbit eye or the chick choriolentoic membrane, which had few or no blood vessels, but put tumors in them and then follow the growth of new vessels to them. But if we were going to have an inhibitor, we would need a delivery system, a tiny little micro or nanoparticle that could deliver any of these molecules we were isolating. And all these molecules we were isolating were big molecules like proteins or nucleic acids or things like that. So at this point, this was 1974, there was almost no work done on drug delivery systems, except for one company had just started, a company called Alza, and Dr. Folkman was on their board. And so he asked them, could they help? But if we go to the next slide, what they said was that it couldn't work because these molecules were just too big. If you encapsulated them in a little particle, they couldn't get out, just like none of us could walk through a wall. So, but if we were going to solve the delivery problem, if we were going to solve the blood vessel problem, we had to solve the delivery problem. So I spent 2 years working in the laboratory, experimenting with different techniques, and I found actually over 200 different ways to get this to not work. But next slide. Eventually, I found a way where I could make little micro or nanoparticles. You see a picture of one on the left, cut in half on the right. And if we go to the next slide, in 1976, we published in Nature the fact that you could use this approach to deliver molecules of any size. This was actually also the very first report that you could use tiny particles to release nucleic acids. Now, I want to go back to 3 months before this. 3 months before this paper came out, I remember giving really the first talk I'd ever given at a scientific conference. I practiced this talk for weeks. It was a 20-minute talk to a bunch of very distinguished scientists, and I was 27 years old. I was very nervous. But anyhow, I practiced for weeks. I got up, I gave the talk, and when I was done, I thought I did okay. I didn't forget that much I was going to say. I didn't stammer too much. So I thought when I was done, all these older scientists, being nice people, would want to encourage me, this young guy. But when I got done, a whole bunch of them came up to me and they said, we don't believe anything you just said. If we could go to the next slide. What they said in particular was, again, the molecules were too big to get out, and the solvents we used, like ethanol and methylene chloride, would destroy whatever we put in. And actually, things went downhill from there. I tried to get research grants, and my first 9 were rejected. The study sections, which were at the health institutes, said that, I didn't know anything about biology or oncology. And then I loved being a postdoc, but a lot of my friends said it's probably not good to do that forever. So I applied for faculty positions in chemical engineering departments. That's my background. And no department in the world would hire me. They said, this bio stuff you're doing makes no sense for a chemical engineer. You should be doing energy or oil or stuff like that. So I didn't get any jobs. Finally, a man in the nutrition department, Nevin Scrimshaw, offered me a job as an assistant professor, but he was kind of a benevolent dictator kind of department head, meaning that he liked me, so he offered me a job, but he didn't bother to ask anybody else in the department what they thought. Now, that might have been okay, except for one thing, which was the year after I joined the department, he left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. In fact, on the next slide, this is from Mike Marletta, who's a very famous chemistry professor, and he was a colleague of mine in 1979. And a number of years ago, I got this award, and he was giving a speech. I'll read a little bit of it to you, referring back to 1979. He said, one evening, I went to a faculty dinner at a Chinese restaurant with myself and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. I always say, but I'm still here, but it wasn't a great beginning. At any rate, we had kept doing this drug delivery work, and we kept understanding it, better and better. And so we were able to actually use it to study the blood vessel inhibitors. And separately, I'd been working on that. I'd isolated maybe 100 fractions, and we're going to test them now in rabbit eyes. I'm going to show you a kind of a gruesome picture, but there's no other way to show blood vessel growth. This is from a paper we wrote in Science, also in 1976. Avastin, which is probably the number 2 or 3 best-selling biotech drug in history, gets approved. And many, many others, as you see on this slide, including 5 last year, get approved. And not only are they approved for all kinds of cancers, but also these are the only pharmacologic treatments that you can use for treating diseases of blindness in the back of the eye, because they also involve abnormal vascularization. Those are diseases like macular degeneration and diabetic retinopathy. Now, going back to the 70s, by the way, the estimate from Nature is that 500 million people will use these. At any rate, going back to the 70s, one of the things that Dr. Folkman and I talked about was writing a patent. And children, I didn't have any patents then, and Children's Hospital in Boston didn't either. So we filed for a patent, and 5 years in a row, it got turned down. And I remember the head of the tech transfer office at the hospital came to see me one day, said, "Bob, just stop." He said, "The examiner, you're wasting so much money. The examiner's never going to allow this." He said, "Just quit." But I don't like to quit. At the same time, science really wasn't working, explaining it to the examiner. But as I told you in the beginning, when we first did this, everyone said it was impossible. It could never work. I wondered, maybe someone wrote that down. So I did the science citation search now in 1981, 1982, looking back at our 1976 Nature paper to see who cited it and what they wrote. Next slide. And I found a number of interesting papers, but this in particular I thought could be helpful. It was by 5 of the top material scientists in the world, and they're describing this field. And here's what they said. Generally, the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example, could be released by such a technique because of their extremely small permeation rates. However, Folkman and myself have reported some surprising, I thought that's a great word for a patent examiner, results that clearly demonstrate the opposite. So we showed that to the examiner. He said, gee, he had no idea, but he said, if Dr. Langer can get written statements, written affidavits from each of these 5 people that they really wrote that, he would allow the patent. So I wrote them, and they were all nice enough to write me back that they really wrote it. And the next slide shows that we got this very broad patent. And with that patent, I got involved in teaching different companies how to do this. We did some licenses. I even got involved in starting companies myself. And on the next slide, what you see are many, many different products based on this. Everything from Lupron Depot, that's Takeda. We had an arrangement with them. They developed these little microspheres that could deliver these luteinizing hormone releasing hormone analogs, which is a peptide, for initially a month, later 6 months, which is what it is today. But many, many others were approved for all kinds of things: eye diseases, pain, opioid addiction, schizophrenia, many things, even for really changing the whole area of aquaculture, which you see in the bottom right, which is now, in part because of this, $250 billion industry, because now for the first time you could control spawning, and even this is releasing GnRH analogs to control reproduction, and it actually enabled preservation of rare fish species. At any rate, so we had done all this, but still, most of these, when you look at them, they're microparticles or injectables. But as we made the particles smaller and smaller, we noticed 2 things. One is that they can stick together and aggregate. 2, that they can be eaten by macrophages. So if you made a really tiny nanoparticle, that might create problems, or would create problems. So on the next slide, what we did is we came up with a way, this was worked with Vladimir Torchelin and others, and of coating particles with polyethylene glycol, which is a FDA approved. It's actually in things you eat. A lot of people drink all the time, like Miralax. And then we even went further and added targeting molecules. This was done by Omid Farrokhzad, so that you could even target things. Let me just show you a video. I don't ever explain this that well, so I thought I'd show you a video that Nova, the TV show, did on our work. Next slide.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
So I should tell you that the clinicians I work with tell me it doesn't really blow the cell up that way, but you get the idea. Now, with this, and actually all through, I mentioned I've been involved in starting a number of different companies. I'll just go over one with you. But this was one we started in 2010 with this idea of nanoparticles. And in this case, making messenger RNA therapeutics. You probably know the central dogma of molecular biology. Now, by 2010, there were a lot of protein therapeutics companies. Protein therapeutics now comprise about 70% of the top 20 best-selling drugs. But, you know, they take a long time to make, maybe a year or two. You have these cell factories or eggs, like for flu vaccine. You start a year in advance with the egg, and you're guessing what vaccine you're going to make against H1N1, H1N2. You don't know. That's why flu vaccines are 40% effective. You're wrong a lot. But we thought if you put, this was done by myself, Nubar Afay and Ken Chen and Derek Rossi. We started this company. It's called Moderna. And the idea is you put messenger RNA and nanoparticles, inject it into the body. And rather than eggs or a cell factory, the body does everything. You just give it the instruction, the messenger RNA. And not only that, next slide, you might be able to treat diseases that you could never treat before, where you're going to make proteins that are intracellular and membrane bound, say for rare diseases. Now, Moderna, actually, the press and stock analysts and a lot of scientists really criticized Moderna. Nature as well, lots of journals did. And a lot of people said this would never work. But the Moderna scientists, of course, I'm prejudiced, a number of them are my former students. They worked really hard. And by the end of 2019, we had 13 products in early stage clinical trials, vaccines and drugs. And then, of course, you know, COVID struck. Next slide. And I wanted to show you what the Moderna scientists did. On January 11th, the Chinese scientists published the genetic sequence of what's called the spike protein. And it took less than 2 days for the Moderna scientists to finalize the design from that and make the mRNA. And then it was put in the nanoparticles and shipped to our clinical partner, NIH, for testing. And the first patients were treated really almost about 2 months after the start of this in Seattle. Now, when you do clinical trials, you always start with what's called a phase 1 trial. That's a small population of patients to find the highest safe dose. So that we started to have data at about 2 months after this, you know, May 19th. And your public company, you have to announce the results. And I thought the results were quite good. It was safe. It got, you know, good antibody titers. I thought finally the press and the scientists and the stock analysts would say good things. But next slide. So this is actually from May 19th, 2020. And the front page headlines of our local paper, the Boston Globe, said, "This is not how you do science." My picture's on the front page. So anyhow, that wasn't so great, but we kept going. You know, you do a phase 2 trial and finally a phase 3 trial. The phase 3 trial, which is the key trial, is a large population of patients. In this case, 30,000. And it was done double blind. 15,000 get a placebo, 15,000 the vaccine. And now in November of 2020, we broke the code, and I'm on the board of directors. So I'm, you know, watching this, and it was a pretty exciting day. I mean, what happened is, and people may know this, but when that code was broken, of those 15,000 who were treated, not a single one died. Not a single one went to the hospital. It was 94% effective. And the next slide shows that, you know, this is a picture of it. This is now approved in over 70 countries. In fact, the only other one that is approved in the US is Pfizer and BioNTech, also putting messenger RNA in nanoparticles. So the idea, this just shows the incredible effectiveness of it between an unvaccinated, vaccinated with a booster or with a booster. And the next slide just shows the effectiveness, which is 94%. Pfizer and BioNTech is 91%. No other vaccine is even near this. And in fact, in the United States, even though over 50 companies have tried to make vaccines. Only these two are on the market and FDA approved. So and it's not just that you can do COVID vaccines. Now RSV vaccines have also gotten excellent data. But what I'm particularly excited about is the idea of personalized cancer vaccines. And this is just from a recent study that was done. This is what's called the phase 2 trial. And the idea of personal, so you probably realize, like most of you get prophylactic vaccines. Those are vaccines that protect you before you get the disease, which the COVID vaccine is. So is flu. But for cancer, you already have the disease. And now what was done is to, in this case, you take 157 patients with a very advanced melanoma. And the way you make this vaccine is you look at the patient's individual cancer profile and you select up to 34 mutations in that patient's cancer cell. And you incorporate the genetic code of those into an mRNA vaccine exactly the same way it was done for the COVID vaccine, mRNA and nanoparticles. And in this trial, half get Keytruda, a very good immunotherapy drug, half get Keytruda plus this vaccine. And it was very, very effective. Risk of recurrence or death reduced by 44%. And normally, and I should say that just recently, the last couple of weeks, the metastases were reduced by 65%. So this, I hope, will lead to many new treatments in the future. Now, I'm going to shift gears a little bit. You know, one of the things that I have just been going over is I did a lot of work on materials. And I also worked at a hospital. And I was curious when I was young, how did materials find their way into medicine? Was it chemists or engineers or some other way? Next slide. And when I looked into this, I found almost always it was medical doctors who were the driving force for getting materials into medicine. And in fact, the artificial heart, that was a surgeon in 1967 going to his house and saying, what has a good flex life for a heart? And he picked a lady's girdle material, and that's how they made the heart. And that's still how it's made today. You know, because once you start down that path from an FDA regulatory standpoint, it's very hard to change. But when blood hits the surface of that heart, the lady's girdle material forms a clot that goes to the patient's brain, and they could get a stroke and die. But if you think about it, something that was designed to be a lady's girdle, you know, it may not be the optimal blood contacting material. And this problem I saw pervaded all of medicine. Now, so we wanted to change this. And so we picked an example. Next slide. And that example, when we started this in 1979, was that at that point, the only FDA-approved synthetic degradable materials, the polyesters, showed bulk erosion, meaning they look like this. Starts out like a phone book, then it gets spongy, and then it falls apart. That's okay for some drugs, but for others, like insulin or an anti-cancer drug, it could be fatal. So we said, from an engineering standpoint, what do you want? And we said, next slide. Surface erosion, like layer by layer erosion. How could you achieve that? So I got chemists and toxicologists in the room, and we put in a blackboard molecules that we thought might work. And if we go to the next slide, I won't go through all of the thinking, but what we did is we tried to analyze monomers, building blocks that might be very safe, and bonds that we thought would break quick enough and would break because of water that you have in your body rather than enzymes, because everybody might have different enzymes. So we did this really big analysis and came up with this structure. And then we synthesized it. And not only did it show surface erosion, but if we go to the next slide, by changing the ratio of those two units that you saw, the CPP and SA, from different ratios, you can make these last anywhere from 2 weeks, like at 79%, to multiple years at 0%. And with that we felt might open the door to other new treatments. And one of those, next slide, that Henry Brem, a young neurosurgeon who's now chief of neurosurgery at Johns Hopkins, brought to our attention, was brain cancer. And in its worst and most common form, that's glioblastoma multiforme. And no matter what you do, the mean lifespan is less than a year. The only drug at this time, when we started working on this in 1984, that was approved by the FDA for brain cancer is shown on the next slide. It's BCNU. But it's a very toxic drug. You give it, goes throughout the whole body and causes terrible toxicity. So Henry and I thought, well, if we could do what's on the next slide, maybe it could work better. And the idea we had is he's going to operate in the patients anyhow as a neurosurgeon. He's take as much of the tumor out as possible, but before closing up the patient, line the surgical cavity with these little wafers, BCNU polymer. Normally, this drug has a 12-minute lifetime, but if it's in the polymer, it's protected, and that should protect it from harm and from degradation. And then you could make it last as long as the polymer lasts. He asked if we could make it last for a month, and you could see from that graph we could do that. So that's what we did. And so that was our idea. Now, of course, if you're a professor, you have to raise money. So we write grants to the government, and they were always rejected. In fact, in 1981, when we first wrote this, the chemists said we'd never be able to synthesize it. After we synthesized it, they said, well, it's going to react with drugs. It's going to react with drugs because of these anhydride bonds. Then they said, well, we keep sending back the grants. They kept saying then, okay, but it's fragile, it won't work. Then that they'd be toxic. Each time we answered, solved a problem, they'd come up with a new one. Then in 1988, they said it won't diffuse far enough. It kept going on until 1996, when actually the FDA approved it. It's the first time in 20 years the FDA ever approved a new treatment for brain cancer. The first time they ever approved this idea of local chemotherapy. You can probably tell from the way I'm speaking that I'm very proud of the work that the students in the lab did. Unfortunately, the reviewers, they didn't do so well. You might ask, how did we ever get the funding in the first place? Well, that was from our patents. We licensed it to a company, Nova Pharmaceuticals, and they gave us the funding to do this. And this, just to show you what this looks like. And again, please don't look if you don't like the sight of blood. But the next slide. I was going to say it's very hard for me to give. That's okay. Go back one. It's very hard for me to give good advice. To get good advice when I give a talk. But many years ago, I was lecturing at MIT to a group of chemists and engineers, and my wife came. And I asked her at the end of the talk, what did you think? And she said, well, the talk, Bob, was okay. That, by the way, is very high praise. But she said, you had those 2 bloody slides on for 10 minutes explaining every detail of the procedure. She said, I don't know if you were looking, but all those chemical engineers were turning green. So ever after that, I did what I did today. I showed it quickly. And I gave a warning. Just to tell you the sequel to that, 2 years later, I was giving a dinner speech. This was to a group of neurosurgeons, though. So I did the same thing. I gave that warning. And at the end of it, a number of neurosurgeons came up to me and they said, Bob, those 2 bloody slides you showed right after dinner, they said, no problem. You could have left those on as long as you want. They said, but those chemical formulas, to leave those on for a neurosurgeon, that's terrible. So I kind of, I hope there's no neurosurgeons in the audience. Anyhow, I just to show you some of the clinical data, this was an early trial and it's not a cure. There's no cure, but this would, you see a 5-fold improvement in survival. And the next slide shows that, this was more recent, where they analyzed 60 trials at 5,000 patients. And again, you get a statistically significant improvement. So this has now been used the last 27 years in over 30 countries. It's no cure. There is no cure for brain cancer, but it relieves suffering and prolongs life. It also opened the door to this idea of localized chemotherapy, which is now used not only in certain types of cancer, but also now in heart disease, where it's had a major impact. Some of this being done by one of my other former students, Elazar Edelman, and also by companies like Boston Scientific. And what I'm talking about here, if we could go to the next slide, is the whole idea of the stent. If somebody has heart disease, probably the most common treatment today for heart disease, when you have really bad heart disease, is to put a stent like this in. It's like a Chinese finger puzzle, keeps the blood vessels open. But the problem is it also causes smooth muscle cell proliferation. And that blocks the vessel and people die and you have to redo it, the stent and so forth. So now what's done is to take Taxol, which is an anti-proliferative drug. Now you can't take it orally because the amount you'd need to get here would kill you, but you coat it onto this stent with a polymer, deliver it locally, and it keeps the vessels open. These are used by millions of patients every year. So I thought I'd end this talk with one other area where we've used materials. And that's to create new tissues and organs. Next slide. So this is a little baby who's dying of liver failure. And Jay Vacanti and I started talking about this in the early 80s. Maybe we could build tissues from scratch rather than treat this child with a transplant where it's done now. Let me just show you our idea on the next slide. This is from a paper we wrote in Science that's been cited about 9,000 times. We wrote this before people talked about stem cells, but you could use those too. But the idea is you could take pretty much any cell type. If you inject it at random, nothing happens. But cells are smart, and so if you put them on a scaffold, they can actually recreate structures. And then you grow them in vitro tissue culture like a bioreactor, and you could then implant these in the patient and make literally any tissue. So as an example, let me just show you the next slide. This is a polymer scaffold with liver cells on it. And a real life example is using this to make skin for burn victims or patients with diabetic skin ulcers. The next slide shows a little baby who's badly burned. But you can take the product, that next slide, which are neonatal skin fibroblasts, put it on the child at the time of injury on the next slide. If you come back 3 weeks later, next slide, he's doing better. If you come back 6 months later, he's pretty much healed. Next slide. Now, not only can you make different tissues and organs, you can also, I think, make tissues and organs on a chip, which could someday reduce animal or human testing. And here, this work that Gordana Bunyak and Milika Ritzik did in our lab, they took cardiomyocytes and in the right media and everything could make, add them together to these scaffolds and make sarcomers and intercalated discs with a cardiac specific ultrastructure. And let me just show you a video of what this looks like. Next slide. This is 6 million heart cells beating like one. We're not shaking this or anything. And what you can now use this for, they even started a company where they do drug testing and they're working, you know, to accelerate drug testing. You could do high throughput screening and you could test many different drugs. I think they're working with about 20 different companies. And you could also even take an EKG as shown on the next slide on this. And I thought I would end this talk by just showing you a picture of all the different tissues and organs we're working with. If we could go to the last slide. And I'm really not going to go through this too much, but we're working on everything from new spinal cords to cartilage to GI tracts, hearing loss, liver. You see vocal cords, where we've actually been working with Julie Andrews, who's who has had damaged vocal cords. You see a picture of me in her there. We're working on brains on a chip, pancreases, and so forth. So thank you very much for listening. I hope that I've given you some examples of how I struggled, but also dreamed, and how you can hopefully use engineering and chemical principles to create things that can relieve suffering and prolong life and hopefully make the world a better place. Thank you so much. I'm happy to take questions.
Fantastic. Thanks very much, Professor Langer.
My pleasure.
Thank you. So we'll now move to the Q&A session. So before we do that, I'd like to introduce my colleagues here, or actually I'll allow them to introduce themselves. They'll be part of the sort of the panel Q&A session that we'll be running.
So my name is Ali Edison. I'm one of the lecturers in the department.
And I'm Camille Petit. I'm a professor here in the same department, Cambridge.
Fantastic. So I guess maybe we can start off and see if anyone have any sort of technical questions. Feel free to raise your hand and ask the question, or you can put it in the chat. If not, we'll ask questions ourselves. So Guybot Stan is going to kick us off. So his question, as you can see in the chat. Oh, have you frozen? Robert, are you with us still?
Oh, can you see? Oh, can you see me?
Oh yeah, okay, yeah, you're there, yeah.
Oh, I'm sorry. Yeah, something must have happened for a second.
Okay, so Guybot Stan asks, when the going got tough, rejections after rejections, what kept you going?
Well, I believed in what I was doing. I just thought that I mean, I guess a couple things. One, I believed in what I was doing. I really thought it could make a difference in the world. You know, also, if I take a look at those two areas in the beginning, you know, I could see these things. I showed you the eye slides. I could see with my own eyes that this was working, you know, that you could really stop the blood vessels. And that meant a lot to me. I also, and I didn't cover this, but when we did the drug delivery work, I put certain enzymes in, those are large molecules, and we developed these assays where I could see a color change. And when I did these, because the enzymes, if they came out, would react with something, and I put 3 different ones in: lysozyme, alkaline phosphatase, and an enzyme inhibitor, soybean trypsin inhibitor. So I was doing these, and I remember when I was doing them, like I failed over and over again. I wasn't kidding about the 200 times, probably even more. And I'd go to and either nothing would come out ever, or it would come out like instantaneously. And finally, I found these designs where came out after an hour, after a day, after 2 days. I kept taking them out of the incubator, and they just kept coming out. So I knew it was working, and so I felt like that made a difference to me. I should say another thing that's always important when you get discouraged is having good friends and colleagues. And I did have that. I had Alex Klebanoff and Mike Marletta and others, and I met Dr. Folkman, and I think I had support, but still it was not pleasant.
Fantastic. Sandra, I think you have your hand up, so feel free to ask the question.
Thank you very much, Bob. It is very inspiring. I mean, very modest as well. I mean, I can just imagine, here, Galileo say, all right, you take a tube, you put a lens, and then you discover pheromones in Mars. Simple. I think. I think that you give an impressive view of things. I'm very intrigued of the fact that how many times have you heard, no, it cannot be done, and yet you went through with it? Many of us, more modestly, still go through and nothing comes out at the other end either. So how much of it is inspiration and how much of it is perspiration?
Well, that's a great question. I think it's some of both. But you're absolutely right. People, especially when you go through the academic review process, and I'm sure it's the same in England, you have these reviewers, and they often can be pretty petty, and even in my case, they still are pretty petty. I mean, I mean, it's funny. Dan Anderson, who started as a postdoc with me and now as a full professor, he always says, like, sending stuff to these review groups is a waste of time. It's like, because they come up with comments that are just ridiculous. Really, they say, well, like, you didn't say what pipette you were going to use. And so I think, so how can we be confident that you can solve this? I'm not kidding. I mean, it's really remarkable what some people can come up with as to why things won't work. And unfortunately, when money's tight, people are very good at deciding why things won't work. So I think it's some of both. I don't think Edison is wrong, about 1% inspiration and 99% perspiration. But, it kind of varies, and I think you need some of both. But there's no question that to get things to the point, certainly where we got with the nanoparticles that are, the idea that you could have nanoparticles that could deliver things as large as mRNA, and of course, many, many other people built on what we did. But I certainly, that's like a close to a 50-year journey of perspiration.
Thank you.
By me and many. We just started.
Thank you.
Great. So Suoro has a question in the chat. Can you share a bit about any difficulties you or your team may have experienced trying to convince investors about funding mRNA vaccines? And how did you address those concerns?
Yeah, well, in the beginning, there were different things. I mean, in the beginning, people, a number of venture groups did say no. And obviously other people had tried to start companies in this also. But you also know that large companies, nobody did it. I mean, really, the only 2 companies that have been successful on mRNA vaccines or mRNA therapeutics, period, are these 2 tiny little companies, Moderna and BioNTech. One in Germany, one in, next to my building at MIT. So I think you keep, unfortunately, Flagship, who started funding the work, Nubar Fayan, who was one of my students. In fact, I think he he may have just gotten an honorary degree at Imperial. I may be wrong, but he is a visionary guy, and he, you know, and I had worked with him for probably close to 40 years, and so he was very visionary, and he was willing to put money in, and we were able to convince others. But it's still, and then we got a really good CEO, Stefan Bancel. I mean, there are people like him who I think deserves enormous credit and leadership, but also he was very good at explaining to people the power that this could possibly have. That doesn't mean it's going to work, but maybe it means to those people that it's worth taking a chance. And of course, the people who invested in Moderna, you know, became very, very rich. I mean, but nonetheless, that's not an atypical thing.
Okay, fantastic. So I think we're going to move on to the questions that people have submitted sort of in advance. So the first question is as follows. So, you know, the biomedical world is really unrecognizable now compared to what it was 20 years ago. So looking forward to the next 20 years, you know, what is your prediction for how it will change? So how different will the world look in 20 years compared to now? So what are your predictions?
Well, of course, the only thing I can say about predictions, whether I make them or somebody else, is they're usually wrong. But that being said, I can tell you what I think might happen or hope might happen. You know, I think we'll certainly see a lot more, what I'll say, therapies based on large molecules, based on genetic therapies, mRNA being a part, but also gene editing approaches and things like that. I think we'll also see maybe more cellular therapies. One of my hopes, and one of the other things that we're spending a lot of time doing, are developing medicines for the developing world. You know, I'd say at least half my lab is aimed, and I have a big lab, so there's a lot of people, are being funded by the Gates Foundation to develop, you know, new delivery systems, new kinds of vaccine systems, better ways of nutrition. And I very much hope that the work we've done, which could enable pills you could swallow to last for a week or a month or the entire course of treatment, vaccines that you can have injected, which have what I'll call self-boosting vaccines. You give one injection, but you may get multiple boosts. New kinds of nutrition that can hopefully, where they might be encapsulated in some ways, but provide vital nutrients like iron or vitamin A that so many millions of people don't get, maybe billions, and many other things. So I hope that all those things will happen in the next 20 years. That doesn't mean we're going to be, and I hope the tissue engineering things that I talked about at the end, I hope that area advances a lot. I'm sure we'll see more things in digital medicine. We'll see things like artificial intelligence probably aiding some of the efforts I mentioned. And I also think, you know, what I always say is sometimes people say, well, what area is going to be really hot, you know, as time goes on? And I said, well, some of the hottest things are the things that don't have a name because they haven't been discovered yet. And I hope they'll be just through basic science, you know, discoveries, things that people have not yet thought about. And the example I always say is you asked anybody 10, 12 years ago what CRISPR was, they wouldn't have known. They'd probably say it has something to do with cooking.
Great, thanks.
Right. So you've built one of the largest biomedical engineering lab in the world. You've said it yourself as well. Do you have any recommendation or tips for people also, you know, at the start of their career trying to build their own research lab or group?
Yeah, well, it wasn't two things. One, it wasn't my intent to build a large one. In fact, I thought by the time now, like I'm almost 75, you know, that maybe I'd be winding down. But of course, I probably never will, because I love what I'm doing. But that being said, you know, the reason, you know, have gotten bigger is because people like Bill Gates and different companies have been kind enough to call me up and say, well, you know, we'll give you funding if you can solve this problem or that problem, and if you'd like to do research in this area or that area. And so, and then I get great people who've applied to our lab, and I don't want to say no to them. That being said, what is the advice? So the advice I'd give to young people starting, first, don't do things that are incremental. I'd rather shoot for doing something that can have a big impact, even if it's hard to do, where you could have great papers and great journals and do something transformative. Obviously, I think it's important to get papers out, But I think it's important to have important papers, and you only live once. I think the second thing I would give advice to is try to raise a lot of money. I mean, I think that money gives you the freedom to pursue your ideas. A third thing I would just say is treat people well. And I'd also say don't give up easily. So those are some of the things that I probably say.
Thank you.
Hello, Professor Langer. So you talked about some of the challenges that you face at the early stages of your career, in particularly persuading the technology transfer office to patent your idea. And you talked about your 1983 patent on controlled release of micromolecules. So how do you know which ideas to patent and when that process starts in your laboratory? And connecting to this question is that, what's your take on open access science, especially for the developing world?
Sure. Well, the second one's easy. I'm absolutely in favor of that for the developing world. You know, we follow what the Gates Foundation's policies are. And that is their policy, even though we publish in good journals. And I think they've worked with those journals to do just what you said. You know, we've continually told them that's important, and they've been great. So that's on the open access thing. On the patent, I consider that different. You know, I think you want to have patents, because it goes back to what we said earlier. You won't get investments in the medical area unless you have patents. No investor will ever put money in anything medical if you don't have the patents, because then they'll say anybody can do it. And when we start, well, I often view a paper like a typical paper we write might have 14 drafts. So a lot of times it might be like draft 3 or 4 you use as the basis of the patent. Sometimes the students want to patent things sooner, and that may be okay too. It really depends how far along you have. I usually like to have a pretty good proof of principle in animals before we write the paper or the patent, but it really varies depending on the discovery. But I think it's always good to have in the back of your mind.
So thank you. Your lab is very well known with its entrepreneurial sort of skill sets. And there was a Harvard Business School case study in 2004, Langer Lab, the Commercializing Science. Referring to this, yourself, how did you develop your personal entrepreneurial identity?
Yeah, well, yeah, there have been a few. Actually, there's a more recent Harvard one. I think the Harvard Business Review has a 2017 article, as well as the case study you mentioned. And I don't consider myself a very good business person or anything, but I learned what I learned by making mistakes, and you know, so and I've also learned one of the things that's important is it's a team effort. You know, I can help on some of the science things, but you really need good business people, good investors, if you're going to make something work. So I think those are some of the things. You know, but it means something to me. You know, I guess I have this passion. You know, I don't want to, and I think a lot of the people in my lab do too. I don't want to just publish something. I want people to use it, and we've been cited a lot. I'm very happy about that. But I also want it to make a difference in the world. And I think that if you don't have patents and you don't have companies, it won't make any difference. Unfortunately, I wish it were the other way.
Right. Moving to the topic of education, what do you see as the changes or trends that are needed to train the next generation of scientists and engineers? Are there things that we need to do less of, or things that we need to do more of, in your opinion?
Yeah, well, I think one thing that's really important is to get the best people to be scientists or engineers. You know, I think so I think popularizing science and engineering is very important. By the way, I think that's one of the great things that England's done, having that Queen Elizabeth Prize, I think is, you know, that was really done to encourage, you know, young engineers about and to try to celebrate you know, science and engineering. But I think that, but that's not something that scientists necessarily can do. I think that, to me, and this is going to sound simplistic, But when I've done teaching, there's sort of been two things. And even when I was mentioning starting the school for poor kids who didn't want to even take math or science, but what I tried to do was two things, and I think they can both be done better in many classes. One is to make science and engineering fun, and two is to make it relevant. In other words, I think if students see that it can do something useful, that makes a big difference to them. That they learn, whether it's taxes for math or like osmotic pills for drug delivery. I mean, so many things. You know, I think if you can show that, build those things into a curriculum, it'll make a big difference. I think you could probably also pay teachers more.
Fair enough. Yeah. Thank
Okay.
you.
So we're going to go back to the online questions. So first, Rory, you have your hand up. Do you want to say your question?
Hi there. Can you hear me okay?
Yeah.
Yes.
Perfect. Thank you so much. Yeah, fascinating talk. Really enjoyed it. So my question is just about how setting up science companies have changed. What are the new challenges entrepreneurs face today that perhaps didn't exist before, and vice versa? Please.
Well, I don't know that it's changed that much. It probably takes more money now, you know, to do things. I also think investors are much more sophisticated now than they were, say, 40 years ago. But I think the general premises are the same. You know, that you come up with some ideas, you get patents, you get some investor money, and you try to make deals for your small company with big companies, not giving away everything, but giving away a little bit that could hopefully give you funding and get a good partner. But I don't think, when I look back over the 40 years, the major thing is probably everything is more expensive now. And investors, of course, have a choice, right? They could invest in a Moderna, which is very capital intensive, because any medical thing is, or they could invest in a Facebook. So one of the things that's important, obviously, is to have laws that, well, you want laws that will, I think, hopefully encourage people investing in medical things rather than discourage them. That, by the way, is why patent laws could be important, and capital gains taxes could be important. And there are also laws that could be negative. So I think that, but the laws obviously change over time, at least in the US, I imagine in England too. So I think that those are some of the things.
Yeah, thanks.
Thank you so much.
So I think actually fittingly we can leave the final question to Omar Matar, who's got his hand up, who's our head of department. So Omar, over to you.
Okay. Yeah, thanks, Yuval. I assume you can hear me okay.
Yes.
Bob, thanks very much for accepting our invitation to come give a talk. I thought this talk was fantastic. Thank you very much. This was the second time I think you've come here. The first one was in person. That was a number of years ago, which you may recall. I've got a couple of really quick ones for you. One of them has got to do with the tenure system in the States. A lot of the work that you do is highly collaborative, right? Do you think the tenure system in the States is still fit for purpose, particularly for the kind of work that you do, where it's a multi-lab type of situation occasionally, and yet if you're going up for tenure, you've got to show your individuality. Where do you think the tenure system is going to be in a few years from now?
Boy, I think that's a great question, and I agree with what you just said. I think there's an overemphasis on really trying to figure out individuality, and sometimes I think it's complicated, because sometimes there's obviously personal things that go on when older people decide about younger people. I could tell you a lot of stories. You probably could tell me a lot of stories. But but the problem is I'm not exactly sure how to change it. I certainly see what you said is 100% right. I just not sure how you change it. I think the only way to maybe change that issue is more leadership, better leadership that says, if people collaborate, it's okay. And you try to really dig into whether all the people made a contribution or not. But it's a great point.
Okay, and then my final question to you is, obviously, you're a successful academic and entrepreneur, etc. Is there anything left for you to achieve? What happens?
Well,
what's the thing that when you achieve, you'll be like, I'm going to have step back and enjoy life? What is that?
Well, the thing is that I always feel like I enjoy life now because I'm doing, I don't even feel that this is a job, you know, I just
Right.
love interacting with the students and you know, coming up with ideas. But I think, you know, so as long as my health is good, I'll probably never do that, you know, so I mean, never step back. But that being said, I mean, what I want to do is, you know, I want to see more of the things that we've started, you know, make a difference to more people. That's why I've been very excited about the work with the, you know, developing world and things like that.
Brilliant. Thanks very much, and good luck with everything.
Thank you so much.
Great. So yeah, we've come to the end of our hour. So thank you once again, Professor Robert Langer. Really inspirational talk, great Q&A. We've really sort of learned a lot, and it was great to have you come virtually, at least, to the Department of Chemical Engineering at Imperial. So on behalf of the department, you know, thank you once again for taking your time to talk to us today. Thanks, and thank you everyone for attending. Thank you. Have a good evening.
Thank you for having me. Take care.
Right.
Bye. Thank you.
Thanks.